Thank you everyone for joining us. This is another webinar in the AUNM series, and it's the second in the chronic diseases or conditions linked to the mitochondria. And we're delighted to have Professor König with us here again.
Hello.
Thank you for being there. I'll just introduce you very, very briefly. She is the Professor of Medical Microbiology and Virology, specializing in infectious immunology and epidemiology. She holds degrees in biology, chemistry, and medicine with a PhD in medical microbiology and immunology. Professor König has served as Deputy Director of the Institute of Medical Microbiology at Leipzig University and has headed the Department of Molecular Microbiology at the University of Magdeburg. She also oversees quality management for both institutions. In 2008, Professor König founded Magdeburg Molecular Detections, MMD, which is the laboratory where AUNM performs its mitochondrial testing, and she serves there as managing director. She's authored 5 books, 9 book chapters, and over 600 peer-reviewed articles. So we're very, very pleased to have her here with us today, and we'll just switch now to the presentation. And it is quite long and quite detailed. We'll probably race through sections of it, so please don't worry. The slides will be made available to you afterwards, and also the recording. And we will go into more detail in a third section of this because we won't have time to cover what comes at the end. We'll be talking about that in a moment. So I'll now switch to it. We'll be talking for about 40 minutes and then have ample time for questions at the end. And all the questions that we don't have time to answer, we'll save and answer next time. So I'll open up the slides now. So the title is The Mitochondria and Chronic Health Conditions, Part 2. And it follows on from the Mitochondria and Chronic Health Conditions, Part 1, that was held towards the end of last year. And this is available on our website, so please do have a look at it. That covered the key tests that, at least certainly M2, as it were, the Mitochondrial Health Index, are vital to do as a foundation. And today we're talking about some supplementary biomarkers that you can do once you've done that one. So we'll primarily be talking about the number of mitochondria, i.e. mitochondrial mass, and reactive oxygen species affecting the mitochondria. And right at the end, we'll talk a little bit about mitochondrial fuel utilization, but we won't have time to talk about therapies for that. So that will be part of our third presentation. So let's dive straight into mitochondrial mass, the ratio of mitochondrial DNA to nuclear DNA. And Professor König, perhaps over to you with this. So thank you.
Yes, you know that mitochondria possess their own DNA. They can't replicate completely alone because the part of DNA or former mitochondrial DNA is now in our nucleus, but there are still mitochondrial DNA in the mitochondria. And if you look on the left side, you see it is around 16,600 base pair long, but you see that there are very important genes on this mitochondrial DNA. And if these genes are not there or not functionally, then the mitochondria can't produce ATP. So this is very important that all these genes or that the mitochondrial DNA is intact. So that is the background first. And here we know, yes, it's okay. And the cell nucleus, yeah, how to determine now it's how do we measure mitochondrial mass? We don't count them in a picture or in a microscope. That would be too much work. We measure by molecular biology the relationship between mitochondrial DNA and the nucleus DNA. It's a technique. You know that you use in the nucleus one gene, which is only one copy there, and you look at your mitochondria, a part or a region which is not mutated, yeah, or which is never mutated, and then you compare the biomolecular biology or PCR and how much copies do you have of your nucleus DNA and how many copies do you have of your mitochondrial DNA. So that is the background how to measure mitochondrial mass. And therefore, we could translate the relationship or the ratio of mitochondrial DNA to nuclear DNA. Here you see a result of 202. Sure, we could translate it into copy of mitochondria because we know that every mitochondria has around 2 to 10 mitochondrial DNA copies. So we can say, okay, that is perhaps around 40 mitochondria. But the ratio is more scientifically in background, and we introduced it, and now it's easier to compare between years before and now. But it's really scientific sound, and so we still use the ratio between mitochondrial DNA and nuclear DNA. And 202 means that in this cell is around 40 mitochondria. And it's the number of mitochondria or mitochondrial DNA, also mitochondrial mass, is dependent on the tissue or on the cell type. And here we know in our peripheral blood mononuclear cells that is the normal range is the ratio of 200 until 250. That is the normal range of mitochondria in peripheral blood mononuclear cells. Next, yeah. And here you see now two different results. Patient 1 has a ratio of mitochondrial DNA to nuclear DNA of 82, and that is very, very low, this amount. It is around 2 mitochondria, 20 mitochondria. But it is a very, very low amount. Therefore, you will see on the right side that is in the red line. And the opposite is you see at patient number 2. There you have a ratio of mitochondrial DNA to nuclear DNA of 1039. And that is very, very high. And you see on the right side that we also have in the red line because if the cell, um, I just mentioned that in the peripheral blood mononuclear cells, the normal range is between 200 and 250 as the ratio of the two DNAs. And if you have very high amount, then is the question, why does the cell need such a lot of mitochondria? Yeah, and if a cell has not enough energy, then first she tries to compensate with a very high number of mitochondria. But later it is a problem because the mitochondria need also energy, nucleotides, lipids, and so on. And then the cell can't offer this again, and then the cells breaks down. Also das that you see, also too low is the top example and too high is the bottom example.
So
Yeah.
there is a way of regulating this. Maybe you could talk about the PGC1 alpha that you also measure in your lab.
Yes, we also measure it because it's very good addition to the number of mitochondria. If you have very, very low amount of mitochondria, then you wish or you need more mitochondria, then you need also an increase in PGC1 alpha. And if you don't have this one, then you know that you have to activate this step or this enzyme. So you can really go better in the therapy what to do. Yeah, if you have very, very low number of mitochondria, but very high PGC1 alpha, then you can say, okay, you are on the right way, but perhaps you need more minerals or vitamins. Yeah. And if you have a very high number of mitochondria and you have still a very high number of PGC1 alpha, then you know that the cell is still needing energy, and that is not the right way the cell is going. So it is very good to have both parameters, the number of mitochondria, also expression as mitochondrial DNA to nuclear DNA, in addition to the expression of PGC1 alpha.
Right. This is just a bit more scientific information on what PGC1 alpha does to induce new mitochondria. I think we won't read through all of this, but it's definitely very, very well established in the scientific literature as being central, and has many, many effects on mitochondrial functions beyond mitobiogenesis as well, which is sort of the full scientific term for that. So it is an RNA test, isn't it? Not a DNA test, this
No,
one
it is
here.
an RNA. It's an RNA test. It's the expression of PGC1 alpha is done. It's by molecular biology, also by PCR, but it's an RNA test, yes. And you always, if you do an RNA test, you have to compare it with a housekeeping gene or the expression of a housekeeping gene. And that is here in this type, the glyceraldehyde 3-phosphate dehydrogenase, or GAPDH, abbreviated. So we always have we have to measure the housekeeping expression of one gene, and then in relationship the PGC1 alpha. That is normal.
Yeah, and this is how it's done worldwide. It's not just a sort
standard,
of
yes.
in-house test.
Yeah.
And of course, if, as we've just seen, the mitochondrial DNA to nuclear DNA ratio is low, and you get a non-detectable or very low result for the PGC1 alpha, then that's definitely a sign that mitobiogenesis would be useful to stimulate exogenously, isn't it?
Yes.
i.e. from outside.
Yeah.
Yeah. In most cases, there are, of course, exceptions. So I'll just try and move on here. So what to do to increase mitochondrial mass or to decrease it if you've got too many mitochondria and they're utilizing energy, but it's clearly, suboptimal to have more than the right number. So first of all, we just want to mention that it's important to see all of this within the overall context of what's going on in the patient. It wouldn't be correct to just say that on the basis of one test, you either, give one or two interventions, or you, remove one or two things and everything will be fine, because you do need to look at the overall what's called often cell danger response, which can be a reason for the mitochondria to have intentionally downregulated, either because of viral or bacterial infections, or perhaps both, or because of biotoxins, mycotoxins. The reasons are that the mitochondria need to sort of go into, they need to downregulate their own activity in order to allow the activities in the cytosol, which is the other part of the cell outside the mitochondria, to fight off what is basically attacking the cell. So this is a well-known phenomenon, which Dr. Navio and others have termed the cell danger response. Parasites can also induce this. Here you can see toxoplasma, which is actually quite a nasty parasite, tethering itself to the mitochondria and disabling it or them. Heavy metals are also vital to look into, because the patient may have a high level of mercury, perhaps from leaking amalgams, or arsenic from the foods they've been eating, the water they've been drinking. There are many, many reasons, even gadolinium from contrast agents used in MRIs. Pesticides and herbicides are also culprits that can block mitochondrial action, and so on. There's a lot else. We won't go into all the others, but we did talk about these a lot more in the first section. So with that context already just mentioned, we can, of course, talk about mitochondrial biogenesis as often a very useful strategy when the number of mitochondria is too low. And that's the growth of new mitochondria and the division of pre-existing mitochondria. And this article, along with thousands of others, explains how that happens. Just in case anybody's interested in the science, we've picked out, I think, one or two that talk about that in detail, but there are many, many out there. Weight is an important factor. Obesity or being overweight can prevent mitochondrial biogenesis occurring as it should. And do you want to say anything about that, Professor König? Why that would affect the number of mitochondria and the ability of the mitochondria to replicate if the patient is, for example, consuming simply too much food? How does that work?
Yeah, that would be a separate meeting in the evening to go through the mechanism.
I know, yeah.
So I would say we can do it another time, because obesity it is influencing the metabolism of the mitochondria at different stages. Yeah.
It's a big, big topic,
Yeah,
isn't it?
yes.
It's just important to keep in mind that that is one issue, especially as nutritional therapists, that we do need to keep in mind. And exercise is important, actually, for giving your mitochondria the signal that they are needed. So bedbound patients will often not have the mitochondrial numbers they should, simply because the mitochondria have sort of gone into hibernation. They've not been given the signals that they are needed. And even after a cardiac operation or whatever, a patient will be given the instructions to exercise a little, and then a bit more and a bit more, just to give their mitochondria the signals: you are needed, please replicate, please start working. And this is a lovely article here with lots of indications of the different boosters of mitochondrial energy that can be used as well. Green tea extract, resveratrol, quercetin. And indeed, resveratrol is well known to stimulate mitochondrial biogenesis. It activates something called SIRT, SIRT1, and PGC1 alpha. And this is a randomized controlled trial where it was found that both these extracts, EGCG and resveratrol, EGCG is the extract from green tea, were able to increase mitochondrial capacity and fat oxidation, i.e. the utilization of fat, to increase mitochondrial numbers. And there's lots and lots of scientific evidence of resveratrol stimulating the number of mitochondria. This is just a few. We do actually have a list of, again, a selection of articles, so do ask us if you'd like that list. PQQ is also able to stimulate metabiogenesis. That stands for pyrroloquinone. And this is, again, another very good, well-researched article that explains that PQQ influences the activity of PGC1 the activity of PGC1 alpha, and is a major mechanism for the regulation of mitochondrial biogenesis. And you will often notice patients, when they take this in whatever combination, perhaps with CoQ10 as well, that it can really, really support their energy. There's another derivative of B3, nicotinamide riboside, and then B3 itself, niacin and quercetin, which are also very helpful. These are further articles that back that up. And alpha lipoic acid. I think, we have pages and pages of articles backing up the action of alpha lipoic acid. Do you want to say anything about that particularly, Professor König? How helpful that can be for metabiogenesis?
No, at the moment not. You see, we have a lot of publications, scientific papers are there, and they show that you can really influence the mitobiogenesis. And perhaps one sentence to the stimuli or to the exercise, you always need an stimulus, whether it's a little bit oxygen radicals, a little bit, or it's, but not too much. That is always the important things, yeah. That you need always, also our cells need stimulus, but not too high. Also too low is not good, too high is not good. You must be in the middle. Yeah.
That's hormesis, isn't it?
Yeah,
yes,
that's hormesis.
absolutely.
Yeah.
Yes. And then we've talked about EGCG, the extract of green tea, already as being very beneficial, not just for metabiogenesis in all kinds of situations, but L-carnitine as well, particularly if the mitochondria are having trouble importing fatty acids. L-carnitine can activate that pathway, and as a result, they may well be able to increase in number where before they couldn't. So that's something that's difficult to obtain if you're a vegan or a vegetarian. It is important to keep in mind how important that is. And copper too, bioavailable copper. This is a lovely article, The Role of Copper in Mitochondrial Biogenesis via its interaction with ATP synthase and cytochrome C oxidase. You'll probably remember from various other webinars we've had, including on KPU, cryptopyrroluria, how very, very important bioavailable copper is for mitochondrial action, and of course, by implication, for the growth of new mitochondria too. This was a title of a very, well read and well-researched magazine called ICAN in the UK. Copper, the Ultimate Mitochondrial Nutrient, was the title of that leader article. And that's because mitochondria can't actually work without bioavailable copper. So their numbers are likely to be low if it's lacking. This is one of, again, many thousands of articles explaining that they are central in particularly complex 4, though they are also important for complex 5, which is ATP synthase that actually makes our energy currency called ATP. And lots of other articles are referenced below explaining that. Photobiomodulation may also have its role, especially near infrared light. There's lots and lots of science coming out now about this. It's really, one aspect of perhaps of the future of medicine. And there are many devices now that you can buy, or big sort of plates that you can purchase to stand in front of, especially if you're an athlete. They can really help with the regeneration of muscle after, long marathon runs or whatever. But this is a small device here that actually doctors in the UK and therapists are using regularly to assist with mitochondrial biogenesis. So I don't know if you want to say a little bit about if we have too many mitochondria, Professor König, just to start off this section. That's called mitophagy, isn't it? It's the opposite, not increasing the number of mitochondria, but decreasing the numbers in a controlled, healthy way.
Yes, and we have in the connection or with our mit unserem Mitochondrial Health Index, you see how much oxygen or maximal oxygen rate you have, also how much is the maximum oxygen consumption rate. And for example, you have 100, and then it's dependent how much or how many mitochondria can can use 100 picomole of oxygen. It is only 50 or 100 or thousands. And if you have a very high number of mitochondria, then you know that the mitochondria by themselves are not functional, not in a healthy condition. And then it's the best. And if you then and if the mitochondria, which are not in a good condition, are replicated again, so you have a lot and more not functionally mitochondria. Therefore, it's a very important step to decrease the number of unhealthy mitochondria. And that is important by the mitophagy. It is also sozusagen the autophagy of mitochondria. So they
Yeah.
must be, the, uh, yeah, the number must be decreased with mitophagy.
So you're just pruning those that are dysfunctional.
Yes,
You're
yes.
not reducing the numbers across the
Not
board.
not the
Exactly.
functional. It's mitochondria have a sensing that they always use dysfunctional mitochondria go into mitophagy.
It's fascinating, isn't it, that they're
Mhm.
able to do that? And
Yes,
certain herbs and remedies are able to assist with that as well.
yes,
The healthy mitochondrial fragments are reused, ideally,
yeah,
aren't they? This is a recycling, as it were. Regulation of mitophagy is explained beautifully in this article here. And so calorie restriction is one way to do that, that
yeah.
has received a lot of, coverage, not just recently, but, over the decades, really. It drives autophagy in a, if it's done in a sort of staged and well thought out way, it can be very helpful. It can prolong life and health span. This is a study about the essential role of autophagy in lifespan extension, using fasting and intermittent fasting as well. But this is often very, very difficult for chronically ill people to do, and not always possible when people are reliant, for example, on intake of fuels from carbohydrates. They need to eat more frequently. You get more calories per mole, from fats. But if they are not able to actually metabolize fats well, it can really be very difficult for patients to do that, as I'm sure you all know who are therapists. So exercise can also be useful, can't it?
Yes.
For
So again,
reducing the number of
yes.
mitochondria as well as increasing the numbers.
Yes, it's the right number, the right dose of exercise you need. Yes, you have again mitochondrial fission and mitophagy, and that is very, very important. So you should go every day a little bit. Don't use the tram for one station. Go uh, and it would be nice to do exercise, regular exercise, several times a week. Yeah, it's
Yeah,
really important.
yeah.
Also, yeah,
It's the
whether you
balance,
like it
isn't
or
it?
not.
It's the balance that's important, if you
Yes,
can. Of course, we know that asking people with severe ME to undergo exercise, graded exercise therapy is not appropriate in many cases. But, this is something that can be discussed with your therapist.
yeah.
So go ahead, sorry.
No, it's okay. Yes, therefore it is for prevention or the new trend with longevity. It's very important. Sure, if you have your problems, you can't do exercise. Yeah, uh,
So we're partly talking about
yeah,
prevention and
yes,
longevity as well, of course.
yeah.
Yeah. So interestingly, there are fantastic articles out there describing the mechanisms and molecular targets of mitophagy triggering phytochemicals. So we're not talking about pharmaceuticals here, but chemicals from nature, and ones that help to prune the mitochondria that are dysfunctional and ensure that they turn over in the healthiest possible way, are again, interestingly, resveratrol, which is able to pick up signals for both mitobiogenesis and mitophagy. So really, if you're just going for one thing, perhaps consider that. And curcumin, And berberine, you'll recognize a lot of these. Sulforaphane, which is from cruciferous vegetables like broccoli sprouts as well. Quercetin comes up again. Tomatidine. So this is a wonderful article, all referenced below, that goes into those in great detail. And there are effects on the nervous system that are very beneficial that you can harvest from these kinds of remedies, like urolithin A, which is from pomegranate. And there are supplements you can find in England as well as in America, where this has been particularly studied, made from pomegranate extract. Of course, drinking it is wonderful too. You just have to find the balance, don't you, Professor König, between actually eating the foods that contain these things and drinking the juices that contain these things, and then taking in too many carbohydrates as a result. You know, fast-acting carbohydrates, and finding that you've upset the balance of nutrients in your system. Sometimes it is a very good idea, particularly for chronically ill people, to access just the extracts of these wonderful herbs. Resveratrol, quercetin, and spermidine are all mentioned here. This here is an article about urolithin A from pomegranates. You can see the picture of them below. And that was a well-powered article with convincing results. We're not mentioning the names of products here, but you know, it's definitely accessible in the UK too now, urolithin A. So it can be problematic to stimulate mitophagy if the person or the patient has too few mitochondria, of course. Look at this test here this result where the person only had a ratio of 2. If they're too weak, in cases of anorexia or amenorrhea, where, you know, a woman who is at an age where she should be cycling is not. Do you want to say anything further about the kinds of medications that may make it difficult for a person to?
Yes, that is again
mitophagy?
How important it is really before just to use things or nutritions or whatever. It is important to know what is the status of the patient. You really to look at the mitochondrial DNA to nuclear DNA. Really, if you have a very low number of mitochondria, still if they are dysfunctional, it would be terrible if you would delete the last existing mitochondria. So it is really important to know where you start. Yeah, what is the basis? What is the background? And then
Exactly.
you can better use all the remedies and whatever. Yeah, so not just give things because it is well known that they are good. No, in this special patient, it can be, yeah, really the wrong way.
Exactly. Yeah. So the balance between the two is very important. Another very good article that explains that, the crosstalk between the two. So now we'll look at the mitochondrial stress test that MMD also performs, which gives a good idea of the reactive oxygen species in a particular part of the mitochondria. I'm sure you will know that mitochondria are the key source of oxidative stress in most cells. It's actually quite a surprise to find that out, isn't it, Professor König? You know, the mitochondria, so beneficial for us, giving us our energy, also produce the most ROS, reactive oxygen species.
Yes, but the ROS are also necessary for signaling, but too much is not good. And therefore, the mitochondria have a lot of enzymes and substances like glutathione, or superoxide dismutase, which decrease the number of oxygen radicals. But a few radicals are necessary for signaling.
Right. And the problem is if there are too many, right? Because
Yes.
numerous diseases are associated with excessive oxidative stress. We won't go through all of them, but really they're involved in every single health dysfunction. You know, they're not the only cause of it, but there's always oxidative stress. And so this is the one measure of oxidative stress that can be performed by MMD. There are others as well. Could you tell us about this one, Professor König? This is called the
Yes.
4977 common deletion, isn't it?
Yes, yes. Also first, if you have oxygen radicals, they first attack the membrane lipids, so you can look at the lipid peroxidation. Then they attack proteins. You can look, and then they attack DNA. And if they attack DNA, especially the mitochondrial DNA, it's a problem. It is this common deletion, as a deletion of the 4977 base pair. It's called common deletion because it's very, it is known for a long time that oxygen radicals lead to the deletion of this part of DNA. And you see that on this part of DNA, of the mitochondrial DNA, are different enzymes which are necessary for ATP production. That means mitochondrial DNA or mitochondria which have mitochondrial DNA with this deletion can't anymore produce ATP. Yeah, and therefore we don't like this common deletion. And there are a lot of wonderful papers, and pathologists have examined that this common deletion is in very high number in your brain and age dependent. Very old people have a lot of this common deletion, and then the brain can't function anymore. And the very old people which are healthy have less of this common deletion.
Oh, okay.
And the second organ is tissue is the heart muscle, where it is accumulating. Yes, it's always tissue, but it's not regenerating very well. So it is accumulating. So it would be nice, or it would be good to never have this common deletion or very low degree. And we measure it in the blood. Blood is regenerative, but if it's in the blood, you know it's everywhere in your body, especially also in the brain. Yeah. And yes, I understand that that's a little bit complicated, the presentation, but we measure how many healthy mitochondrial DNA in relationship to mitochondrial DNA with this common deletion. Yeah, and there we have, if you have this number, it means that only 10 to the high of 10
To the power of.
To the power, thank you, to the power of 6 are healthy, and one has the common deletion. Yeah, and we must be higher. Also there must be the power of 8 or more. The power of 8 must be is good. Yeah,
It's essential. Yeah.
essential.
This number, I mean, it's always shown on the bar, isn't it?
Yes, yes.
And luckily, you're
on the
always
bar,
available
yes.
to speak to if we need further explanation. By the way, she's very available, which is lovely. But in this case, the oxidative stress was noticeably enhanced along the mitochondrial genome, and that's quite serious, isn't it? As it
Yes.
says at the bottom there, this is a direct quotation. This can cause complete failure of ATP production in the mitochondria affected. So what's important is, again, to prune out those mitochondria, isn't it? It's important to renew them.
Yes, and there's also another problem, because if mitochondria, you must delete this mitochondria with this common deletion, because mitochondria with this common deletion are divided faster. Yeah, also because they are shorter, the DNA, and that is a further problem.
Okay, so getting on to if I can switch to switch to the next page. There we are. Action can be taken. It's not as if this is permanent. We wouldn't want to measure any polymorphisms that couldn't be altered with these tests. And actually, you can, can't you, improve the situation of oxidative stress. So this just backs up what Professor König told us, that it can be found in all parts of the body, that it does reflect other parts of the body, right? It's not just representative of the peripheral blood mononucleosides, which we're pulling out of the blood. So I'll go on to the next page now. And EMFs, that's electromagnetic frequency, is a major cause of oxidative stress. So particularly in patients who are chronically ill, this can make a big, big difference to help protect them from it. The reason is that the mitochondria are little electromagnetic, organisms of their own. Their mitochondria have a particular voltage gradient, their membranes, which have to be functioning well. And unfortunately, this is something that Professor Martin Paul found many, many years ago and has written about with big teams since, that the voltage-gated calcium channels can open up, particularly in chronic illness. Calcium flows in. This is driven partially by EMFs, and it can really lead to severe dysfunction in patients. So something to consider. There are lots and lots of things you can do to reduce that, and that would be a presentation all of its own, really, which we do have, and we work closely, for example, with Dr. Dietrich Klinghardt, who talks about that a lot, and there are talks of his available. Heavy metals can induce oxygen radicals too, so to keep those in mind and to reduce them to the extent you can, or at least to look into where they could be coming from, and then to reduce your exposure. Mercury mentioned there again. SARS-CoV-2, COVID, has unfortunately created havoc in the mitochondria, because it has, as a very clear part of its mechanism, created reactive oxygen species. And this is what is happening in some cases of long COVID. This is one of, again, many hundreds of articles explaining this, and the mitochondrial membrane depolarization that results. So you do want to keep that in mind with long COVID. Hyperglycemia is also a driver of mitochondrial oxidative stress. And so you want to look at the patient's blood sugar markers too, and just make sure that you don't have metabolic syndrome developing. Of course, if the patient has diabetes, then you're in a very particular situation that needs factoring in. And iron overload, something else to consider very, very carefully, something that long COVID and the last few years have accentuated, unfortunately. So mitochondrial iron overload promotes reactive oxygen species. You really want to get that balance back again, reduce that overload that may well be evident in the iron markers in your patient. And cryptopyruria, which we've talked about a lot at AONM, this glitch in the production of heme is also associated with oxidative stress, as this article, among many others, explains. So medications too, this is just one of many that can cause severe oxidative stress in the mitochondria. So we're talking about removing as well as adding. It's not just about adding, antioxidants in the diet and in the excellent supplements that exist, but it's also about removing anything that could be causing this disruption of your mitochondrial function. So we can look at the NRF2 antioxidant regulator, can't we, Professor König, in order to know where we're going with oxidative stress. Could you maybe talk us through this a little bit?
Yes, a little bit. We have seen that oxidative stress, how to treat oxidative stress. Sure, we can give antioxidants, but we will look how is the cell regulating oxidative stress. And there's one master regulator, it's NRF2, also as in nuclear factor erythroid factor 2 related factor, also NRF2 we say, and he regulates a lot of other enzymes. So we don't know exactly what the cell needs, but the NRF2 then exactly knows what to do. It's activating enzymes which destroy superoxides. They are activating enzymes which downregulate or destroy or inactivate heavy metals or pharmaceuticals and so on. So the NRF2 is really a master regulator, and it is very important to activate NRF2. Yeah, then we can say, okay, NRF2 knows what to do with, but we need to look at NRF2. And it's also a good factor to look whether there is an expression. For example, if you have high oxidative stress, for example, a high number of mitochondria with the common deletion, then it's very important to know, do you have NRF2 activation? Or do you have a problem that your cell doesn't activate NRF2 and can't defend against the oxidative stress?
And this is something you can measure as well. Let's just have a
Yes,
look at
that
the
we
test.
can measure.
Here we are.
Mhm.
Another RNA test, isn't it?
Yes, it's an RNA test, although again, messenger RNA, and therefore you have again a reference gene, which you measure the mRNA and you compare the expression. And in this case, it's not detectable. Again, if you have in parallel a very high oxidative stress, like a common deletion, high number of common deletion mitochondrial DNA, then this is not a good situation. Then you have to try not to activate the NRF2, and perhaps you can now add a little bit antioxidants until you have activated your NRF2. Yeah, so different
Which
steps.
for which there's lots and lots of evidence of ways to do that. Now, we don't have time. I see we're already, roundabout question time right now.
Time
So
is
we don't
running.
want to go into the detail too much, but we do have lots and lots
Yeah.
of articles that we've linked to of what you can do to activate your NRF2. Just to mention, it is important also on the Mitochondrial Health Index, which we talked about last time, to check where else you might have oxidative stress showing. Because even if you don't have oxidative stress that's actually beginning to compromise your genome, which is the most serious form, isn't it? You may, on the page 2 of the Mitochondrial Health Index, see increased formation of oxygen radicals in the cell or increased formation of oxygen radicals in the mitochondria. That's along the electron transport chain. So don't assume that just because your patient doesn't have this 4977 deletion showing that they don't have ROS. Most cases of chronic illness, which is what we're talking about as practitioners, do exhibit that, don't they, Professor König? It's very, very unusual not to have reactive oxygen species in the body, isn't it?
Yes.
In the mitochondria. So how to quell that, we'll just mention that the articles are here. We won't go through them in detail. This is one with a long, long list of, often you'll find supplements that do contain many, many different antioxidants, and they're often composed of exactly these kinds of things like reishi and quercetin and curcumin and sulforaphane, rosemary and sage. Carnosol is the ingredient in those. This broccoli extract again contains high amounts of antioxidants. Olive oil too, the extract from that, oleonic acid. A list here that looks very complex and detailed, but actually if you look down the left there, orange juice, apple juice, beetroot juice, it's not that unusual. These are just wonderful antioxidants too, and do activate NRF2. And these ones here, of which we have many, many more sources, so we can let you have them if you'd like them, are all very, very well researched with human studies behind them, not just, animal studies. And some of these you will have seen. Vitamin C, we haven't actually mentioned. Melatonin, lactoferrin, glutathione, we have mentioned. Even hydrogen-rich water. So glutathione, this is a lovely article by Ben Brown, our very own. He runs the Nutrition Medicine Institute, and they have wonderful events as well. He wrote this article together with Dr. Minich about where you can access glutathione and how much to take. and a few other remedies that perhaps we'll go into in greater detail next time. This is an unknown one in the UK called dragon's blood. It seems to have the highest oxygen radical absorption capacity of anything else, over 2 million. And whereas blueberries, blackberries have around about 3,600. So an absolutely extraordinary remedy that is much better known in Germany than here, from a tree in the Amazon. It's the sap from a tree and has been packaged into antioxidant supplements. Many, many others. So we won't go through them in detail. Just to say that this test of mitochondrial oxidation levels can also be done as a finger prick test. So that's very, very useful for children, for patients who are bedbound and where you can't draw blood. What we'll do is we'll leave the last part, which is about mitochondrial fuels, I think, Brigitte, until next time. Is that okay?
Yeah. Yes,
because we've
okay.
talked in such detail about those first two, and I expected that. So we'll talk about how to check for the mitochondrial fuels that your mitochondria are actually able to use and what to do about the results next time. So we'll leave those last few charts. And if you haven't seen part 1 yet, that's under mitochondrial webinars, and it's the middle one here. And so I'll stop sharing now and just see whether we have any questions in the chat or the Q&A. So, oh my goodness, yes, we do have a lot. I'll just go to the beginning of the chat and see how many we can cover in the short time. I'm sorry about that. We went through everything in perhaps a bit too much detail, but there's so much to say.
Sorry if I explained too much.
No, I was talking a lot as well. I mean, it needs to be in several different sessions because there's so much information about the mitochondria now that's been coming out, particularly over the last decade. So I'll scroll down here and just because of talking, I wasn't able to check them through. One question here for you, perhaps, Brigitte. How do you prioritize interventions when multiple deficits are found? For example, low mitochondrial number and impaired fuel flexibility. How do you decide where to begin?
Yeah, that is a very good question. Where to start? Sometimes I think perhaps it's good to start first to know what is the person, what is the lifestyle. Perhaps you start with some infusion or a very good vitamins and so on. So they have everything, yeah. Just to do it for 2 weeks. And then look, perhaps it's only one substance missing, and then it's good again. Also this is, I would start, and then to look, can you introduce a little bit exercise or not? Or if you look exactly at the Mitochondrial Health Index, you can see a little bit where to start at the antioxidants, enough too, or whether you have to go more in supplements and so on. You can see it a little bit. Yeah, but first I would say start with a broad supplementation, perhaps addition with infusions, just to have a background, yeah, because everything what you are doing needs that the cells can do what you want.
Absolutely. I mean, infusions is perhaps something we should have a separate session about and pull in some of our doctors here from the UK who give them. I think that's an excellent point. One question here is, would you advise giving liposomal supplements such as glutathione for increased reactive oxygen species? Is it better to use a liposomal form?
It depends. For me, it is always better that the body is producing it by himself. So it must have cysteine, glycine, as one example. And if you give a normal glutathione or glutathione? Glutathione. Then it is degraded before it enters the cells. That is normal. And inside the cells, it must be put together again. And if your cells doesn't, or if these enzymes are not functioning well, then you can't get glutathione, either if you give a lot of glutathione through infusion or or as a supplement. And in these cases, sometimes it would be good to start with liposomal glutathione, just to be sure that it's going in the cells. But then I would say it's better to look that the enzymes are functioning well. You can do it with some need selene, some need zinc. Also it is, yeah.
Understand. Thank you.
Mm.
Would taking D-ribose increase ATP sufficiently to increase mitochondrial function?
It's not sufficient alone.
Not alone.
Not
Yeah.
alone.
Yeah. But it can be helpful, can't it?
Yes,
Especially
sure.
for de novo production
Yeah.
of ATP when you're completely exhausted. If someone's going through active cancer treatment, the treatments are generally oxidative, and therefore where in the cycle would the optimum time be to test mitochondrial function?
Yeah, cancer is a very difficult topic. And I think it's not too complicated just to give a short answer. But if you want to measure, it depends. Do you want to measure the mitochondrial function of, for example, T cells, which are relevant for the defense against tumor cells? Yes, and it would be nice just not using the totally peripheral blood mononuclear cells, but to go exactly only to the T cells because it's their function. And then we can see how the mitochondria are functioning there. So I would say in a cancer patient, I would really go on specialized cell types.
Yeah, well, maybe we'll have a separate session together with you and Professor Pachmann, who does our cancer sort of liquid
Yes.
biopsies.
Mm,
That would be
yes.
fascinating, wouldn't it? And
Yeah.
talk about, because it depends very much on the half-life of the
Yes.
immunotherapy as well,
Yeah.
doesn't it? So I'll just go to the Q&A now. Sorry that I spent all that time on the chat when there's lots in the Q&A as well. Are there tests available at the professor's lab which can determine which pathogens are active and affecting the system? What we normally do is combine those with Armin Labs tests, which we also supply, as you'll know. You do also do tests of pathogens, don't you, Professor König? But I know you're more specialized in the mitochondria. And
yes, we at the moment we didn't because we have a lot of things to do with the mitochondria.
yeah,
But
yeah.
yes, I like microorganisms. I did a lot with parasites and viruses and bacteria. So it is, and we did a lot of studies also in Africa. So it is important to know exactly whether you have a reactivation or not, yeah, or persistence. And if you have a persistence, it's also important. Yeah, it's difficult to know where are the bacteria or whatever hiding. Yeah, where are they? Yeah. So you can, for example, if you want to know whether Borrelia are somewhere, yeah, perhaps you can take a very high number of blood and then there are specific techniques that you isolate only bacterial DNA to be very sensitive or to go in this direction, and that we will now focus on this part.
Thank you. There are quite a few questions asking whether your tests are available in the USA. We do tests for some doctors and therapists in the USA, and the difficulty is making sure that the samples reach the lab fast enough, because for the foundational tests like the Mitochondrial Health Index, intracellular ATP, they do need to reach the lab within 48 hours, maximum 60. So it depends really on where in the USA your patients live and whether the courier system is fast enough. But we are succeeding quite often, aren't we, Professor König? It used to be more difficult, but we are now managing, aren't we, to get
Yes,
samples from the
they need
USA.
your help. I'm just measuring, analyzing the cells. Also, USA is the problem of the transport, but I think there you are doing a great job.
We're managing, yes. So
Yes,
please
yes,
do ask. Our
yes.
helpline, that's info@aonm.org, can give you all the information on that, the exact specifications for how to transport it if you have a courier of your own that you'd like to suggest using. Are there certain people who should not take quercetin? That's an interesting question, isn't it? I believe that is the case with COMT. That's a COMT variation in your genes. That can be problematic. Do you know of any other reasons why people shouldn't take quercetin?
No, at the moment, not.
No.
No?
There was a question right at the top here that from Mr. Lindfors. Is it, I'll read it. I haven't had a chance to absorb it yet, so it may be too complex for us to answer quickly, but is it worth taking a copper supplement if you've got a deficiency of ECU and the metabolomics urine test shows that there's too much copper in the urine? It seems that not all copper is absorbed and it perhaps lacks some other components to react with. This seems to be the case with many EDS patients. That's fascinating. Copper utilization and the balance of copper and iron in the body is a massive topic. I'd have to look into that, Mr. Lindfors. I can't immediately give you an answer to that. And of course, the metabolomics urine test is very useful for showing levels of trace minerals and metals, but I wouldn't be able to give a direct answer to that. I don't know if you feel you can, Brigitte, give an answer to that.
No, it is like with everything, it's complicated. It's always the balance, and I think we have to go deeper in this topic.
Yes, we may well have a separate talk all about iron, copper, and the mitochondria. Yes, I'd love that, actually. We have time for maybe one or two more questions. Let me just see. Perhaps the last question here. Professor König, how should you divide your daily antioxidant intake if you've decided that it is the right thing to take antioxidants? Would you take them before or after exercise?
Because I do moderate exercise, it doesn't matter. I have not, but I really take it before and some hours after exercise. But it depends how much exercise you have. You need a little bit stimulus. That is always important. Yeah.
Yeah, I think that's something that would be important to discuss with your physical trainers, for example, or your therapist, depending on the type of exercise and your physical constitution. Sorry.
And therefore, I like also to activate the nerve too. Yeah.
Absolutely. Yeah. Make sure that that's activated. Well, I think we've reached the top of the hour now and need to close this session for now. But we'll be saving all the questions and everything in the chat as well for next time. Thank you so much, everyone, for being there. And Professor König, thank you
for
all your time,
for listening,
knowledge. Just to mention that there is another webinar coming up on the 17th of September at 6 p.m. That's an Armin Labs training. And you can register for that. The registration link is in the chat. And we also have a webinar with Dr. Sarah Myhill on the 7th of October called Water, the Single Most Important Molecule for Life, how it impacts on energy delivery mechanisms and inflammation with implications for management. So that will be fascinating as well. And there's a conference next week being held in Finland that I'm actually going to, called the First Annual Conference on Chronic Infectious Pathologies. And I'll come back with information from that, lots and lots of presentations and new ideas. So I'll give an update on that either in the newsletter or in a webinar. And we do also have ILADS, the International Lyme and Associated Diseases Symposium, holding their next conference in London for the first time next year. So lots and lots ahead. Thank you, everybody, for being there. And again, Professor König, thank you so much for spending your time with us today. Thank you.
Good night.
Night.